Hello everyone, in this video we are going to discuss what is southern blotting. As you know, southern blotting is a technique which we use to separate the DNA fragments. The southern blotting, it was a technique which was developed by a scientist called Edward Southern. That's why it's called southern blotting. So the principle behind the southern blotting is same as the principle behind gel electrophoresis. So DNA is negatively charged. If we look at the structure of DNA, we know that there is a phosphate group present in the fifth carbon. So there is a PO3 minus group present in the fifth carbon. This negative group, this PO3 minus group, which is present in the fifth carbon, will give the whole DNA a negative charge. This particular group is responsible for the negative charge or this property of our DNA. So what we do is we take advantage of this property of DNA to force it towards a positive terminal. Okay, so DNA is negatively charged. So if we apply a potential difference in an agarose medium, it will move towards the positive electrode. So if I have an agarose medium and I provide a potential difference, the DNA fragments which I load here will move towards this end, this positive end. So DNA is negatively charged, this end will be positive. So the DNA which I insert here, which I load here, will move towards the positive electrode because of its negative charge. And how exactly does it move? The agarose shell has minute pores inside it. So the DNA moves through that pore. So it has minute pores inside it. So DNA basically moves through that pore. Okay. But here we are discussing about southern blotting, and we are going to see what is southern blotting in detail. So as you know, we are basically separating DNA fragments. So in order to get the DNA outside, what do we have to do? DNA will be seen inside a cell. Okay, let us say it's a prokaryotic cell, so we don't need to show any nucleus. So DNA will be present inside the cell. If we have to get the DNA outside, we first have to break the cell membrane and the cell wall if it is a prokaryotic organism. So basically we have to break this thing. And how exactly do we break this cell membrane? We know that cell membrane is made up of lipids. Okay, we could use the normal dishwash which we use in our kitchen. How? The lipids present in the dishwash will go and compete with this lipid which are seen in this cell membrane. Okay, this lipid from the detergent. Okay, it will go and compete with this lipid molecules, and it will generate a pressure, and eventually the whole thing will break. We could use even dishwash, we use other chemicals also. So basically we use uh, something which is having the property of dishwash to break this cell membrane and cell wall. We could use STS also to denature all the proteins. Our purpose is to break the cell wall and cell membrane and get the DNA outside. So now it's broken. I have the DNA and everything inside of the test tube. Now I just want my DNA or my nucleic acid. How am I going to do that? I am going to add STS or something which will denature the proteins, and I will add a proteinase enzyme also which will digest all the proteins. At last I will use phenol or chloroform mixture to phenol or chloroform or its mixture to get my DNA. Okay, so I have my DNA. This is how I extract my DNA from. This is the basic principle behind the sorry, this is the procedure of extraction of DNA. So now we are going to delve into the actual topic, southern blotting. So here what we are going to do is, we have the DNA and we will lyse the cell to get the DNA outside, and we will convert it into fragments. Okay, using our various nuclease enzyme, we will lyse it into fragments. Okay, now this DNA fragments, so the first step is fragmentation. So once we get the DNA fragments, we are going to go for gel electrophoresis. Okay, so I have the gel electrophoresis set up here. I have prepared the agarose shell and everything. So now I am going to load my DNA sample into the wells here. I will apply a potential difference of 50 to 100 volt. Okay, I'm going to load the DNA into this well. Now what happens is, when I apply the potential difference, this DNA fragments will start moving towards this positive electrode. Okay, smaller fragments will be able to move through that pores easily, and it will like move faster, and it will be seen somewhere here, like, imagine. But the larger fragments will not be able to move that fastly, so it will remain somewhere over here. And what just happened is, they separated according to their size. Okay, they have separated according to their size. Now, next, what I am going to do is I will um, soak them in an alkaline solution. Soak in an alkaline solution. Next, what is going to happen is I'm going to put this gel on top of a uh, like a solid support. After that, I will apply uh, a pressure or a vacuum to get this DNA outside. Okay, and I will transfer it into a nitrocellulose paper. Now, once we soak it in alkaline solution, next what we are going to do is we are going to get the DNA outside. We are going to transfer it into a um, nitrocellulose paper. So next, how exactly do we do that? We'll apply a negative pressure here so that the DNA will come outside, and yeah, once we get the DNA fragment, we will transfer it into a nitrocellulose membrane, and next what is going to happen is uh, we are going to hybridize it. We got the DNA outside, and we are going to add a probe DNA. What is a probe DNA? A probe DNA is a known DNA. We know the DNA sequence of that particular DNA fragment. We know the uh, sequence of this fragment. Okay, so this is our probe DNA, and we are going to hybridize this two. But this uh, probe DNA was labeled with fluorescently labeled with radioactive uh, isotope. So this probe has a fluorescent tag. It is labeled with radioactive isotopes. So what is going to happen here is uh, what is going to happen here is during the process of hybridization, this DNA here will form a duplex, meaning they will form bonds. Okay, and this one, this imagine this is our probe DNA, and this one is fluorescently labeled. Next, what is going to happen is we will go for washing. That is, all the things on top of all the unhybridized uh, thing on top of that nitrocellulose will get washed off. Then we are going to go for detection. Okay, here we know that we have labeled it with radioactive components. So this probe has radioactive components. So when using autoradiography, we will be able to visualize this DNA. So this one is hybridized with this one. If there is a mismatch or say some portion is not emitting any signal, from that we could say that there is some kind of mismatching over here. Okay, this is what Saturn